Okay, so welcome back to this next video in which we are discussing phospholipase C beta enzymes. Okay, right, so we've now seen that there are these 4 genes within the phospholipase C beta enzyme family, and that they, 3 of them have multiple splice variants, giving overall 7 known phospholipase C beta enzymes. Okay, right. So what we now want to discuss is the structure of phospholipase C beta enzymes. So I'm going to start off with a picture of the primary structure of a phospholipase C beta enzyme, and then we'll sort of build up to trying to draw a three-dimensional picture, and then we'll put this all together to try and understand how these things are going to be activated by the alpha-Q/11 subunits of heterotrimeric G proteins. Okay, right, so we're drawing a primary structure firstly then. So we'll start off with the amino terminus of the polypeptide here. So all of these phospholipase C beta enzymes are a single polypeptide. They're one protein, okay? So you don't have to stick multiple polypeptides together to make a phospholipase C beta enzyme. It's just a single polypeptide. So here is its amino terminus. Then the first important domain that you have is something known as a plextrin homology domain. Okay, and this is going to be semi-important in binding phospholipase C beta enzymes to the phospholipid bilayer, well, to the underside of the phospholipid bilayer. It's not as important as it is in other phospholipase C enzyme types, okay? So in phospholipase C beta family enzymes, the plextrin homology domain doesn't play as crucial a role as, for instance, it plays in the phospholipase C gamma enzymes. Okay, so this is what's known as a plextrin homology domain, or at least it's representing a plextrin homology domain. Okay, so what does this mean? Well, basically, it's a type of domain that you have in many different proteins, okay? So it's a certain conserved structure that you find in loads of different proteins, and originally it was found in this protein called plextrin, which is a protein found in platelets, okay? And every time we've seen it in another protein, we've then called it the domain that is homologous to that domain that we found in plextrin. Okay, so we call it the plextrin homology domain. And for short, plextrin homology domains are just abbreviated to PH domains for plextrin homology domain. Okay, right. So as I say, that domain is going to be important in binding us to the underside of the phospholipid bilayer. It's going to be semi-important. Okay, it's not the only thing that's going to be involved in that. Okay, after that, what you have is 4 EF hand domains. Okay, and these types of domains, usually you find them in things like calmodulin. Calmodulin is the famous protein that has 4 EF hand domains. Okay, and in calmodulin, these EF hand domains are the domains which bind calcium. However, in phospholipase C beta enzymes, these 4 EF hand domains are not going to bind calcium. So it's not fully understood what the significance of these 4 EF hand domains being here is. Okay, so we're not going to see a massive role for these in the function of the phospholipase C beta enzymes or the activation of them at all, really. Okay, so, but they are here. They are these 4 EF hand domains, as they're called. Okay, I'm going to have to start making things smaller, otherwise I'm not going to fit everything in. Next up, we have a really important set of domains. Okay, these are the domains which are actually going to form the catalytic site. Okay, so you have 2 of them. You have what's known as an X domain. Okay, so this is called the X domain. And then you have a little linker between the X domain and then the Y domain. Now, in other forms of phospholipase C enzymes, such as the phospholipase C gamma enzymes, this linker, which is called the XY linker, is much, much bigger, and it has a huge plethora of different structures within it. Okay, in the phospholipase C beta family of enzymes, it's not that large. So I'll colour in the XY linker in green here, and then we'll colour in the X and the Y domains in yellow. So here's the X domain in yellow, and here's the Y domain in yellow. Okay, and we'll see those, we're going to study the structure of these in a lot more detail in a moment, but for now just know that together they're going to dimerize together and form the catalytic site, the site that is actually going to be involved in converting phosphatidylinositol 4,5-bisphosphate into diacylglycerol and inositol 1,4,5-trisphosphate. Now, after that, you then have a C2 domain. Okay, now C2 domains are also incredibly important in calcium signaling. Like EF hand domains, C2 domains are one of these other famous domains which binds calcium. And you know, you have C2 domains famously in protein kinase C enzymes, and that's how calcium is involved in activating protein kinase C enzymes. Okay, but again, in the phospholipase C beta enzymes, the C2 domains do not even seem to bind calcium, and they don't seem to be that important in activating the enzyme. In other phospholipase C enzymes, the C2 domain plays a much more important role, but in the beta family of phospholipase C enzymes, it's not important, or at least we don't think so. Okay, now comes an extremely important domain, okay, which isn't even going to get a box. In fact, I probably should give it something. Okay, I'll try to. Here, this box here. Okay, this is known as the C-terminal domain, and this is going to be really important both in the interaction with the alpha Q/11 subunits of heterotrimeric G proteins, and also with regards to binding to the underside of the phospholipid bilayer. So this is what's known as the C-terminal domain. Okay, and for short, the C-terminal domain is abbreviated to the CTD. Okay, right, so that's the primary structure then of a phospholipase C beta family enzyme. Okay, before we can discuss how this is going to form a three-dimensional structure, there is something that I need to discuss with you first, which is the concept of a TIM barrel. Okay, so basically, the X and the Y domains, which are going to make up the catalytic sites, the catalytic site rather, of our phospholipase C beta enzyme, these are going to form a domain known as a TIM-like barrel domain. Okay, and in order to understand what a TIM-like barrel domain is, we firstly need to discuss what a TIM barrel domain is. Okay, so we're now going to discuss what a TIM barrel domain is. Okay, and then we'll see how the X and the Y form something that's almost a TIM barrel domain. Okay, so what does TIM stand for here? TIM stands for triose phosphate isomerase. Okay, which is an enzyme important in glycolysis. Okay, it turns dihydroxyacetone phosphate into glyceraldehyde 3-phosphate, if my biochemistry serves me right. Okay, and basically this TIM barrel domain was first found in triose phosphate isomerase, and hence it was called a TIM barrel domain. Okay, so T for triose, and then IM for isomerase, IM. Okay, right. So they have another name. They're also called alpha-beta barrels, which in some ways is a better name than TIM barrel domain. They like the TIM name because it, forms TIM, but alpha-beta barrel is probably a better name. So let me discuss the structure of an alpha-beta barrel with you now. So basically, what you're going to have is alternating beta strands and alpha helices. Okay, so what's going to happen is within your polypeptide, what you're going to have is a structure where you have a beta strand, which is just a straight polypeptide like so, which is going to interact with other straight polypeptides via the hydrogen bonds between the peptide links and alternating ones, but we'll come back to that. And then you're going to have an alpha helix. Okay, so here's an alpha helix. And you're going to continue that alternation another 7 times. Okay, so I'll only draw this once. I'll color-code it in future so that we can draw the alpha helix looking like a spring. So here is the alpha helix, but of course the alpha helices do look like springs, but I'll color code them in future and then just get away with doing it like that. Okay, so overall in one of these tin barrel domains or an alpha beta barrel, you're going to have 8 beta strands which are all going parallel, because as you can see they're all going upwards. Okay, so they're all beautifully parallel, and these 8 beta strands are all going to interact with each other and form a beta pleated sheet in a way that I'll show you in a moment. Okay, here again is our alpha helix, and then one final one here. Okay, right, so what I'll also say is this is the amino side. Okay, I'm not going to put the amino terminus there because there could be other structures here. Indeed, in this case, there are these other structures here because it's the XY domains which are going to form something along these lines. Okay, so this would have the amino terminus. Sometimes people put something like this. They'll put the amino terminus there, and then they'll put 2 lines like this to show, there could be something in between there. Okay, so this is the amino side, and then similarly on this side, this is the carboxylic acid side, but again, there could be a lot more in between there than just the carboxylic acid terminus being instantly after this. Okay, also let's color code this now so that I can get away with not having to draw it quite the same way again. So here in orange we'll show all of these parallel beta strands. Okay, and all the beta strand is a nice straight portion of a polypeptide which can then interact with another straight portion of the polypeptide, and they can all form a beta pleated sheet together. Okay, so these are beta strands, and they're all parallel to one another. They're all going in the same direction. Okay, so their amino termini are all down here, and their carboxylic acid termini are all up here. Okay, now let's colour in the alpha helices in red, which doesn't look a great colour. I should have picked something more different from orange, but never mind. Okay, so we then have these 8 alpha helices. Okay, so we've got alternating beta strands and alpha helices. So this is why it's called an alpha beta barrel. I haven't shown you why it's called a barrel yet, but at least now you can understand where the alpha beta comes from. Okay, so now it's time to show you where the concept of a barrel comes from. Okay, and it would be nice if I could fit this in on the same page. I'll try down here. Okay, so basically this whole structure is going to fold up into a barrel-like structure. Okay, so you're going to get basically something that looks like this. A cylinder-like shape with a hole going down the middle. So the whole thing is going to fold round into this barrel-like shape. And the beta strands are going to be on the inside of the barrel, and the alpha helices are going to be on the outside of the barrel. Okay, so let me show you this. I'm going to draw this picture from above now. Okay, so if we draw it from above, like so. Okay, so you've got this portion of the protein, and then you've got a channel down the middle, effectively. Okay, what you find is that the beta strands, which are in orange, those are all on the inside of the barrel. So you find 1, 2, 3, 4, 5, 6, 7, 8. All 8 of the beta strands are on the inside of the barrel, whilst the 8 alpha helices are then on the outside of the barrel, like so. 1, 2, 3, 4, 5, 6, 7, 8. And tin barrels like this, or alpha beta barrels, they usually do have 8 beta strands and 8 alpha helices. Okay, so starting this off then, you start with your amino terminus here, and I'll use this symbol again to denote that you could have a lot in between this first beta strand and the amino terminus. Okay, then you've got this first beta strand, then you'll go into your alpha helix here, so this is how the polypeptide goes. Then from that alpha helix you'll go into this beta strand, this alpha helix, this beta strand, this alpha helix, this beta strand, etc., like so. And that's how the entire thing is folding up. And then you'll end with this alpha helix, and then you'll go off into the carboxylic acid terminus like that. Okay, so that's how you're folding these 8 beta strands and these 8 alpha helices together to make this alpha beta barrel, or this tin barrel. Okay, so that's the sort of structure then that you have in triose phosphate isomerase. Okay, you have a similar structure within these phospholipase C beta enzymes, and in fact you find this in all of the phospholipase C enzymes, but we're studying phospholipase C beta specifically. Basically, that X and Y domain, that consists of portions like this, where you've got alternating beta strands and alpha helices. Okay, and I'd just like to stress one more thing before we move on, that these beta strands here, they're all interacting with each other, and effectively they're forming a nice beta-pleated sheet there. Okay, it's wrapped around in a cylinder-like shape, but it is a beta-pleated sheet. Okay, right. So, going back to our phospholipase C beta enzymes, the X and the Y domain are going to form portions like this, okay? And together, they're going to come together and form a barrel-like domain, a tin barrel-like domain. Now, they don't quite form a complete tin barrel. They don't quite have the 8 beta strands and the 8 alpha helices, but they form something pretty close. So let's draw this structure then now. Okay, so let's then start off by drawing our tin barrel-like domain here. Okay, so I'll draw my barrel here, and of course it's got the cavity down the middle there. Okay, and half of this is going to be made up of the X domain, and then the other half will be the Y domain up here. Okay, and then there's a linker between the two, and the linker, this XY linker, folds down into that cavity between the X and the Y domain within the middle of the barrel. Okay, so let's colour in these different portions. I wish I had the other primary structure on the same piece of paper. That was badly planned. Okay, so here then is the X domain here in yellow. Okay. And we then have the Y domain behind, and together they are forming this tin-like barrel domain here. Okay, with that XY linker between them, connecting them up, that is then folding into this cavity between the two that's formed in the middle of the barrel. Okay, so this is an XY-like, well, this is the XY linker rather. Okay, so this doesn't, as I say, form a complete tin barrel. You don't quite have 8 beta strands and 8 alpha helices, but it gets pretty close. Okay, so now let's draw the rest of the polypeptides. So before the X domain here, what we had was those 4 EF hand domains here. So this is the X and the Y, and we've drawn the XY linker so far. We now need to add on the things that were before the X domain, and then the things that were after the Y domain. Okay, so starting with the things which were before the X domain. Okay, so before the X domain, you have these 4 EF hand domains, which we don't really know what they're doing. Okay, so here they are anyway, though. So we have our 4 EF hand domains there. So I'll colour those in purple again. Okay, but remember, they don't bind calcium in the case of the phospholipase C beta enzymes. Right. Then, after the 4 EF hand domains, what we then have is that plextrin homology domain, which is going to play a role in binding to the phospholipid by their underside in phospholipase C beta enzymes, but it's nowhere near as important as it is in other phospholipase C family enzymes. Okay, so here's the amino terminus of our phospholipase C beta enzyme, and then the first domain that we have after the amino terminus is here in turquoise, and that's the plextrin homology domain. And this is going to play a role in binding to the phospholipid by there, but in, for instance, other phospholipase C enzymes, such as phospholipase C gamma, this is absolutely horrendously important, whereas in phospholipase C betas, its role in binding to the phospholipid by there is sort of minimal, basically. Okay, but it does have some role. Then after the Y domain, what we have is that C2 domain, again, which is far more important in other phospholipase C beta and phospholipase C enzymes, but isn't too important in the phospholipase C beta enzymes. Okay, it doesn't even bind calcium in the case of the phospholipase C beta enzymes. And then after that we have our C-terminal domain here, which is extraordinarily important. So this is the C-terminal domain. And then we finally got the C-terminus itself. There. Okay, right, so this then now is a three-dimensional cartoon of our phospholipase C beta enzyme. Okay, right, so let's discuss which portion then is the catalytic domain. This cavity that's within the XY barrel here, which is the TIM-like barrel domain here, the cavity down the middle, this is the catalytic domain. What's going to happen is phosphatidylenoacetyl 4,5-bisphosphate molecules are going to stick into that cavity. Okay, so I'll try and draw this here. That was a bad idea, it's messing this up. Okay, but never mind, I've got to go ahead now. So here is our here's our phospholipase C beta enzyme with our PIP2 molecule here. What's going to happen is it will stick into this cavity its head here, its head consisting of the inositol ring, and I'll draw it out again. The inositol ring here with the free phosphate groups coming off, okay, so the IP3 molecule in effect, or the IP3 molecule to B if you like. Okay, so it's going to stick all of this into that catalytic domain, and the catalytic domain is then going to cut the bond between the glycerol molecule, specifically the third carbon of the glycerol molecule, and that phosphate group that comes off the alcohol group of the third carbon of the glycerol molecule. Okay, so let's colour this in. Here are the phosphate groups again. Okay, here is the inositol ring in blue, and then here are the long chain carboxylic acids in orange. Okay, right. So, in order for the PIP2 molecule to stick its head into the catalytic domain, you firstly have to remove the XY linker. The XY linker is stopping the XY domain, which is this TIM-like barrel domain, from actually allowing a PIP2 molecule into the catalytic domain, basically. Okay, so you firstly have to move the XY linker out, then PIP2 molecules can stick their heads into this catalytic core here, and then the reaction will be catalyzed. Okay, so firstly what I want to discuss is the membrane association of these phospholipase C beta enzymes. How do they bind to the phospholipid bilayer, and then we'll move on to how the alpha Q/11 subunits of heterotrimeric G proteins are going to activate them. Okay, right, so basically the plextrin homology domain and the C-terminal domain, these attach to the underside of the phospholipid bilayer. Okay, so if this is the cell membrane here, this is the inner leaflet, this is the outer leaflet, so this is the extracellular fluid on this side, and this is the cytoplasm on this side. Okay, and what's going to happen is the C-terminal domain and the plextrin homology domain bind to the inner leaflet of the phospholipid bilayer in ways that still aren't fully understood. Okay, but what we do know is that the C-terminal domain is far more important for this than the plextrin homology domain. Okay, and what happens is you continually have an equilibrium. Okay, continually phospholipase C beta enzymes are falling off the cell membrane and then rebinding onto the inner leaflet of the phospholipid bilayer, falling off, rebinding on, so you continually have this falling off the cell membrane and then rebinding. So at any one time you'll have some phospholipase C beta enzymes that are attached to the inner leaflet of the phospholipid bilayer and some that are not. Okay, so it's continually moving between being just floating around in the cytoplasm and being attached to the inner leaflet of the phospholipid bilayer. So let me show this again. So here is our C-terminal domain now bound to the underside of the inner leaflet of the phospholipid bilayer. In addition, we need to somehow show our plextrin homology domain bound, but I'll worry about that in a moment. Okay, so here's our C2 domain, here's our C-terminal domain. Okay, so we're working along our protein in the opposite order to which is conventional. going from carboxy terminal to amino terminal rather than from amino terminal to carboxy terminal. Okay, and the idea is that at the moment, even when it's bound to the inner leaflet of the phospholipid bilayer, the tin-like barrel isn't going to be properly in contact with the phospholipid bilayer. Okay, so at the moment, even though the phospholipase C-beta enzyme is moving between the cytoplasm and the underside of the phospholipid bilayer, even when it's attached to the underside of the phospholipid bilayer, it's not active at the moment. So we're talking about the inactive enzyme. The inactive enzyme can be moving between the cytoplasm and the underside of the phospholipid bilayer, and it's not active at either one of those states. Okay, we'll talk about how you activate it later. So what will happen is the XY domain here, which is this tin-like barrel domain, will not be properly positioned to interact with the phospholipid bilayer. Okay. In addition, you'll have the XY linker blocking the catalytic site. Okay, so here is the XY linker in purple here. Okay, so this is the X domain and this is the Y domain here. And then coming off the X domain, you then have the 4 EF hand domains, 1, 2, and I was trying to show the plextrin homology domain bound to the phospholipid bilayer, right, forgot about that. We're going to have to stretch this out a little bit, okay, here we have another EF hand domain and another EF hand domain there, and now let's have our plextrin homology domain also bound to the underside of the phospholipid bilayer, and then we'll have our amino terminus. Okay, so that's a little bit of artistic license there to show that both the C-terminal domain and the plextrin homology domain are involved in this binding to the underside of the phospholipid bilayer, and I'll stress once again that this one is more important. Okay, right. So we continually have the enzyme moving from being free in the cytoplasm to being attached to the underside of the phospholipid bilayer, but in both states it's still inactivated at the moment. Okay, what can now happen is the alpha Q/11 subunits of heterotrimeric G proteins can activate the phospholipase C-beta enzymes whilst they are in this membrane-bound state. Okay, and we'll see that in the next video.